If you've ever seen Hollywood sci-fi movies, you've probably seen this, the twisted double helix of DNA. Scientists might further depict it like this, or this. But to clearly explain the purpose of DNA in Cell City, we shall depict it like this, and remove the twist. DNA is similar to RNA. Both molecules are composed of nucleotides, linked together in linear chains. And though DNA contains a nucleotide called thymidine rather than uridine, the nucleotides of DNA and RNA share many of the same chemical properties. An obvious difference between the two is that DNA is composed of two chains rather than one. Chains that stick tightly together because of the chemistry of DNA nucleotides. Remember how a messenger RNA can attract and stick to a transfer RNA? Through complementary base pairing. It is complementary base pairing between T and A and G and C that cause the DNA strands to stick together as well. In DNA, the T's of one strand fully complement the A's on the other, while its C's match up perfectly with its G's. As a result, the two single strands of DNA stick tightly together and move around as one. DNA is a long linear molecule. Stretched out like this, it couldn't possibly fit inside Cell City. So it is packaged into a more compact form and stored within the nucleus at the center of the cell. This chromosome is a single coiled DNA molecule. More advanced humans like yourself contain 46 chromosomes within each cell nucleus, but the cell of Anatom stores only one, which will make it easier to study. Let's unpackage the chromosome to see its DNA tightly coiled around protein spools for efficient storage. When uncoiled and stretched to its full length, DNA once again appears as a single linear double-stranded molecule. But this time, notice the segments of DNA that are highlighted in green. These are gene regions. Some protein citizens are quite actively involved with genes. For example, RNA polymerase. RNA polymerase separates the two strands of DNA within a gene region and reads one of the strands. Watch what it accomplishes. RNA polymerase creates a chain of RNA nucleotides, block by block, following the instructions given to it by DNA. A review of nucleotide chemistry will show how this is possible. Remember how RNA nucleotides are chemically attracted to each other in certain pairs? And that DNA nucleotides display complementary binding as well. The same attraction occurs between RNA and DNA nucleotides, because RNA and DNA are chemically very similar. Here we have placed the RNA nucleotides above the DNA nucleotides to show that complementary pairing occurs between U and A, G and C, C and G, and A and T. For every DNA nucleotide in a gene, RNA polymerase binds an RNA nucleotide complementary to it and uses the energy within that nucleotide to link it to the end of a growing RNA chain. Now, as you might expect, RNA polymerase requires energy to accomplish these actions. And once again, ATP is there to provide it. Isn't it fascinating that the main source of energy for all activities in Cell City also serves as a building block for RNA and DNA? 3 huge jobs performed by one small molecule. After reading a gene sequence of DNA, RNA polymerase releases the RNA strand it has created. As it is shuttled out of the nucleus, it becomes messenger RNA. We know the fate of messenger RNA once outside the nucleus. It is fed into the ribosome and translated into an amino acid chain. In this case, the last chain needed to complete this shuttle protein complex. The RNA polymerase, which we shall now simplify for clearer viewing, will continue to read genes and build RNAs from them whenever more protein citizens are needed by Cell City. Since accidents are always happening there, and proteins are often damaged, Cell City must take measures to maintain its protein population. So it employs other protein citizens to keep a head count and stimulate the production of protein citizen replacements when necessary. The DNA of Cell City contains coding regions called genes. Genes are the instructions for creating protein citizens. Protein citizens read genes to create more of themselves and to replace fellow citizens who have become injured or destroyed.